This is part 3 of the cellular structure lectures. We left off with this slide. We've talked about lysosomes and how lysosomes contain digestive enzymes. They're only found in animal cells. Plant cells don't have lysosomes. Any digesting, and I'll tell you the reason is because animals eat food, and that food is in all sorts of forms. I mean, just think about what you ate for dinner last night. Maybe it was spaghetti. So you've got pasta, you've got the ground beef, which has a mixture of protein and fat, and then you've got your tomato sauce. I mean, maybe you sprinkled some Parmesan cheese on it. But anyway, the point is we eat a large variety of food, and so plants don't do that. They make their own food, and they don't make a large variety. So they once they make their food, they don't really need to digest it. It's already produced in a digested form. So lysosomes are needed by animal cells for that reason, because we do eat a large variety of food that has to be broken down. So this is showing you, this picture here is showing you a white blood cell. It actually looks a lot like a protozoan called an amoeba. I think I've seen this picture in my Bio 112 class as well. But white blood cells and amoeba are very similar. So it doesn't really matter if this is a white blood cell or if it's an amoeba. It's got these extensions called pseudopods. And the pseudopods can wrap around a food particle or a bacteria and bring it into the cell, and now you have a food vacuole or a vacuole that's storing your bacteria, and the lysosome will come over and fuse with that food vacuole, and those digestive enzymes will break down that food vacuole or that bacteria, and then it will fuse with the plasma membrane of the cell, and it will carry out exocytosis and secrete the waste outside the cell. Chloroplasts are the structures inside of plant cells only. They're plant cells, animal cells do not have chloroplasts. Chloroplasts carry out photosynthesis, which is where they use energy from sunlight to convert carbon dioxide into carbohydrates. And the carbohydrates is their food. So they make their food from carbon dioxide and sunlight. They have their own DNA and ribosomes. So again, there's a theory that chloroplasts were photosynthetic bacteria at one time that were engulfed by a larger cell, and they lived symbiotically for many years until finally that photosynthetic bacteria just became the chloroplast, a constant part of the eukaryotic cell. It's showing you that the chloroplast has a double membrane, an outer and an inner membrane, and that it's also got these little discs called thylakoids, and then each stack of thylakoids is called a granum. And it is the thylakoid of the chloroplast that actually has the chlorophyll pigment in it. It's the part that makes it green. So the thylakoid contains the chlorophyll. And the light reactions where sunlight is captured by the chloroplast, those occur in the thylakoid. And then the reactions called Calvin cycle reactions, which are when the food is actually produced, those occur in the stroma. The stroma is just the fluid. All right, vacuoles are membrane-bound sacs that function in storage and transport, but they cannot fuse with the plasma membrane. They cannot fuse with the membranes of other cellular components. So vesicles can, but vacuoles cannot. In plants, the central vacuole is large, and it plays a key role in regulating a plant cell's concentration of water in changing environmental conditions. So it stores a large amount of water for the plant. The cytoskeleton is a network of protein fibers, 3 different types of protein fibers, and they have all different functions. Microfilaments, which you see here, thicken the cortex, or the outer edge of the cell. In other words, they're just inside the plasma membrane. If this were a cell, the plasma membrane would be here. Okay? All right, and they're like rubber bands. They resist tension. Microtubules are found inside the cell. They help maintain the cell's shape. Microtubules are the thickest of the protein fibers, and they're going to play a large role when the cell divides in cell division. Microfilaments are the thinnest, and then intermediate filaments are kind of in the middle as far as their thickness, and they hold the organelles in place. The microfilaments function in cellular movement. They can disassemble and reform quickly, so that can help the cell to change its shape and also to move. What you're looking at here is a filament called actin, which is very important in muscle cell contractions. The intermediate filaments help hold organelles in place and maintain the shape of the cell. The microtubules are the largest of the protein fibers, and they can also dissolve and reform quickly. Okay, so microtubules are a component of, I was wanting to talk to you about the, I don't know if it's on here or not. No, it isn't. Okay, let's go back to the picture of the centrosome. Okay, so what you're looking at here is a centrosome, and a centrosome is an organizing center that helps organize the chromosomes when the cell divides. It helps to line them up and then separate them so that each, when the cell divides by meiosis and produces, sperm and egg, each sperm and egg gets half the chromosomes. Or if the cell divides by mitosis, each new cell gets a copy of what the old cell had. In animal cells, the centrosome includes centrioles, and the centrioles contain these microtubule triplets. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. I think I'm counting 9. Anyway, there's 9 of these groups of triplet microtubules. So microtubules form the centrioles, which are going to play a role in cell division in animal cells. And then let's go back here. They also produce flagella, which are hair-like structures that function in movement. In humans, a flagellum is found on each sperm cell. Cilia are shorter hair-like structures, but both the flagella and the cilia have the same arrangement of, what you can see here is 2 microtubules in a pair. And if you look, there's 9 sets of 2, 5, 6, 7, 8, 9. And then there's 1 set of 2 in the middle. So we call this a 9 plus 2 arrangement of microtubules. And this is what you see both in the flagella and the cilia. The plasma membrane of a cell, sometimes called the cell membrane, we have a whole chapter on it. Unit 5 is on the plasma membrane. It's mainly composed of a double layer of phospholipid molecules. Each one of these molecules that you see that look like this with the red heads and then the two yellow tails. These are called phospholipids. And there is a double layer of phospholipids in the plasma membrane, a phospholipid bilayer. It's embedded with proteins, which are the large blue molecules that you see. And then sometimes what you see on the outside are carbohydrates attached. If they're attached to the phospholipids, then we call this a glycolipid. If they're attached to a protein, we call it a glycoprotein. So glyco just refers to a carbohydrate chain attached. This goes back to the Golgi complex. Once the lipid or the protein arrives at the Golgi complex from the endoplasmic reticulum, the Golgi complex can add a carbohydrate chain to it and make it a glycolipid or a glycoprotein. Some cells have folded plasma membranes, and those folds are called microvilli. We have cells like this lining our intestines, and what it is like if this were the inside of your intestines, if you were to just take a cross section of the inside of your intestines, and everywhere that lines the inside of your intestines absorbs nutrients that are produced from breaking down the food that you eat. But what if the inside were folded? You see how much more lining that gives us? So folding increases surface area or absorption. We can absorb much more nutrients having microvilli than we can if it's just a flat surface. All right, plasmodesmata are junctions between plant cells. They're junctions between plant cell walls so that different chemicals, it's like a channel, different chemicals can pass from one cell to another. Animal cells have cell junctions as well. They have junctions called tight junctions, desmosomes, and gap junctions. And then okay, I was getting ready to say, they are going to give us pictures, right? The extracellular matrix, that means what is outside the cell, is the tissue and chemicals that enable the cell to attach to other cells and communicate with other cells. So this is whatever's outside the plasma membrane, that's called the extracellular matrix. All right, gap junctions in animal cells are channels between cells. They're connections, but they form a channel. So they're, right here it shows you a little bit better. This is a channel between, this would be one cell here, and this would be another cell. And what it allows in the heart muscle cells, it allows ions to move so quickly through cardiac muscle from one cell to another that the entire heart contracts instead of the muscles contracting separately. And tight junctions are like a tight seal. You have tight junctions lining your urinary bladder because you do not want there to be any way for urine to escape into your body. Desmosomes are very strong connections that form between cells, kind of like a spot weld. It's like one button-like connection, but it's really strong. It holds tissues that stretch a lot need desmosomes. If you think about it, if you are lighter skinned and you've ever been sunburnt, and later on the after effects are that you shed some skin, you peeled your skin off in sheets. It didn't come off one cell at a time. And that's because our skin cells are held together by desmosomes. That's a very strong connection that holds our skin cells together. And also, if you've ever had dandruff or if you've ever had skin that flakes off, sometimes called ashy, that if you look at those flakes, they're actually, you can see them, so you know they're large and they contain more than one cell, and that is because the cells are connected by desmosomes.